Hello, everyone. It's Tuesday, June 22nd, and I just want to talk about the video transport take two, specifically the different types of gated channels. So here we can see this is going to be a ligand gated channel because the ligand or signaling molecule will bind to this spot right here to open up this gate. So a couple things I want to emphasize. When we have gated channels, they are allowing something to move in. So for example, if we look at this gated channel, which would have been that gated channel we were just looking at, but the ligand is now bound to that channel, the gate is open. Let's pretend this signaling molecule in orange right here, which is the ligand or signaling molecule, let's pretend that's acetylcholine. Don't worry too much about what acetylcholine is right now, but it is a neurotransmitter that we will talk about quite often this semester. Acetylcholine is a signaling molecule that opens up a ligand-gated ion channel. The ligand is never what is passing through that channel. That is to say, acetylcholine does not pass through the channel. Acetylcholine is merely the key that opens up this door, if you will. So we would call this a ligand-gated sodium channel because it's going to open up the gate, and because sodium always wants to move passively into the cell down its gradient, sodium will then do so. What I've talked about in the past is that ions or molecules will move down their gradients if given the opportunity. So if we look at the channel right here, the gate is closed. Here we can see the gate's closed because there's no there's no ligand bounded to that channel. So sodium is not being given the opportunity to move down its gradient into the cell. If a ligand binds to it, in this case, we'll just say it's acetylcholine, then sodium is given the opportunity to move into the cell. So one thing I want to point out, the ligand is never the entity that's moving through the channel. It's merely unlocking the door, opening the door, allowing the specific ion or molecule to move through. It could be potassium. Potassium certainly would be moving the other direction. In the insulin release video, we see the potassium ATPase channel, which ATP is the ligand in that scenario, and potassium is the entity which moves through the channel down its gradient out of the cell. In this case, sodium moves down its gradient into the cell. So these gated channels, the ligand is never moving through the channel. And please keep in mind, whatever is moving through the channel is still doing so passively. It does not require energy to move through the channel. All that it needs is that gate to be open. So gated channels do not require energy. The entity, in this case sodium, is moving passively into the cell. Okay, that's it for this little update. I will see you guys 3 o'clock today for office hours, Tuesday, June 22nd.